Next up, we have Yuta Matsuoka from Kyoto University. So Yuta is an assistant professor from Kyoto University Graduate School of Medicine. Throughout his career, has advanced research concerning the development application of lipid peroxidation analysis techniques. He's actually published the work he's going to present today in a preprint of the study, which you can read on the BBCon portal. And the title of his talk is Volatile Oxidized Lipids Generated via Metal-Dependent Lipid Peroxidation of PUFAs of Breath Biomarkers for Monitoring Ferroptosis in Vivo.
Hello everyone, I'm Yuta Matsuoka of Kyoto University. In this presentation, I'd like to talk about our developed breast biomarker for monitoring the ferroptosis in Vivo. As you know, the human exhaled breath contains more than 1,000 types of volatile organic compounds, VOCs. These molecules are produced through the various metabolic reactions. So the amount and the types of VOCs change depending on the health status and the disease onset. For instance, the exhaled aldehydes are elevated in the cancer patients. On the other hand, these hydrocarbons are found in the exhaled breath of patients with liver cirrhosis. As shown here, the profile of exhaled VOCs depends on the type of diseases. So there are expected to be non-invasive biomarkers of several diseases. However, many challenges still remain for the proposal of VOCs as disease biomarkers. The one such challenge is the lack of understanding of their origins. That is the generation mechanism of each VOC. To date, many VOCs have been identified in human exhaled breath. However, in most studies, it remains unclear whether they are actually involved in the pathogenesis. So this may be the big limitation of biomarker development. And to address these challenges, we are taking a different approach from the previous VOC studies. In our study, we first identified the VOCs produced by the specific metabolic reaction based on the in vitro experiments and clarified their generation mechanism. Then, using the developed library of these VOCs, we applied it to discover disease biomarkers. In this study, we focused on the lipid peroxidation as VOC formation reaction. The lipid peroxidation is one of the primary sources of biological VOCs. The membrane lipid, especially those containing the polyunsaturated fatty acids PUFA, are easily oxidized and fragmented by the intercellular loss and iron. Through these reactions, volatile oxidized lipids (VOLs) such as hydrocarbons and aldehydes are produced and released from the lipid membrane. Furthermore, lipid peroxidation leads to the membrane rupture and ferroptosis. This is a type of the cell death dependent on the lipid peroxidation. In recent years, many reports have shown that this cell death, the ferroptosis, contributes to the various diseases. So, in other words, the analytical techniques for the VOLs can contribute to the development of breast biomarkers for ferroptosis in vivo and ferroptosis-related diseases. Therefore, this study aims to develop the screening method for ferroptosis-related diseases through the breast analysis, targeting the volatile oxidized lipid released from the ferroptotic cells. So, however, the detailed generation mechanism of volatile oxidized lipid are not clear, and only a few types of these molecules have been identified. So because of this, it is unknown what kinds of peroxidized lipids are produced from the ferroptotic cells and released from the exhaled breath. To overcome this issue, in this study, we first constructed the structure library of VOLs using the GCMS-based analysis. Furthermore, we applied our developed library to identify the VOL released from the ferroptotic cell and its related disease model animals. First, we conducted in vitro experiment using the standard lipids to construct VOL structure library. These lipids were oxidized within a sealed vial, and the VOL released into the gas phase were analyzed by the GCMS combined with solid phase microextraction method. This heat map represents the abundance of the VOLs derived from the arachidonic acid and deoxyxyanoic acid. Each VOLs increased by adding the oxidizing reagent APH and hemine. Interestingly, the profile of VOLs were totally different between the arachidonic acid and the DHA. So this is because the VOL cleavage reactions originate from the omega terminal structure of the fatty acid. And furthermore, we also found that the different VOLs can be produced from the same lipid depending on the type of oxidation initiator. For example, in the metal-independent oxidation, a large amount of these aldehydes is produced. On the other hand, in metal-dependent oxidation, the more vinyl alcohol are produced. These results suggest that the VOL profile can be used to predict the precursor lipid and the mechanism of oxidation reactions. Finally, we identified the 48 VOLs derived from the 4 PUFAs and constructed the structure library. Using the constructed library, we attempted to identify the VOLs released from the ferroptotic cells. The hepzid cells are used for this experiment, and the ferroptosis was induced by the RSS3, that is the inhibitor of the GPX4. As a result, the 4 hours treatment of RSS3 induced the LDH release and oxidized lipids formation, and these are suppressed by co-treatment of the RIP1, that is the inhibitor of lipid peroxidation and ferroptosis. So we next corrected and analyzed the cell-derived biogas at this time point. The 4 hours treatment RSS3, the cells are pressed into the sealed virus and their biogas were measured by the SPME-GCMS method. As a result, several VOLs, especially the 1.30 and 2-pensulfan, show the significant increase by RSL3 treatment. Furthermore, this increase was suppressed by the RIP1. And also, similar results are observed in the case of using the other cell lines and other ferroptosis inducers such as FINO2 and ML162. Importantly, these molecules were not released during other forms of cell death such as apoptosis. So based on these results, we conducted further investigation on the VOLs as a candidate for ferroptosis gas biomarkers. Next, we attempted to clarify their generation mechanism. Actually, these molecules were observed only in the oxidation of the omega-6 PUFA, especially arachidonic acid. So we next conducted tracer experiments using the stable isotope level lipids to validate whether the VOLs is produced from these lipids during the ferroptosis. By the treatment of isotope level lipids to help G2 cell for the 3 hours, and they are incorporated in the membrane host lipid. Then, after inducing the ferroptosis, we measured the delabeled VOLs produced. So as a result, when HepG2 were treated with the delabeled arachidonic acid and the renoic acid, the very high amount of the delabeled VOL were observed. So this result indicates that the biomarkers is produced from the omega-6 PUFA containing lipid during the ferroptosis. This is a plausible generation mechanism of the 1-octane This molecule can be formed through the vascularization of the 12-HPT by the Fenton reaction, followed by the intermolecular cleavage. Indeed, when several lipid peroxides were mixed with a metal ion, the 1-octane was detected only when using the 12-HPT. Furthermore, we are also able to detect these intermediates produced by this mechanism. Then we next examined whether VOL marker can be detected in biogas released from the ferroptosis-related disease model animals. As a disease model, we initially used the acetaminophen-induced liver injury model, the mice. The acetaminophen APP is a fibro-reducing medication frequently used in the clinical settings. However, in cases of the overdose, hepatic glutathione is depleted and the ferroptosis and acute liver damage occurs. So in this study, we found that the liver damage and the production of the oxidized lipid, this is a signature of the ferroptosis, dramatically increased in the mice treated with APAP. And the liver damage were suppressed by the treatment with N-acetylcysteine. This is a substrate for the glutathione synthesis. So following that, we analyzed the gas molecule released from these model mice. So here, I'd like to show you the experimental setup to correct the VOS emitted by the mice. The mice were placed within a sealed flask, and the 2 tubes are passed through. One supplied clean air, and the other was connected to a thermal desorption tube to capture the VOS. Then it was analyzed by the TDGCM system. Here you can see the increase of these VOLs in biogas of APAP-treated mice, and it is suppressed by the NAC treatment. Then, to investigate whether these VOLs are actually produced in the liver, the amount of hepatic VOLs were measured. As a result, the production of hepatic VOLs increased with APAP treatment. So these results suggest that VOLs can be used for the non-invasive measurement of ferroptosis in vivo. So following that, we further examined whether this marker could also be applied to detect the ferroptosis occurring in the chronic diseases. In this study, we focused on the metabolic dysfunction associated with steatohepatitis, the MASH. The MASH is a chronic liver disease characterized by the hepatic steatosis and inflammation. Since the 20% of NASH patients progress to the end-stage liver diseases such as the cirrhosis and the hepatocellular carcinoma, so our diagnosis is quite important. But the gold standard for MASH diagnosis is liver biopsy, which is a highly invasive method. So the development of MASH biomarker is awaited. And here, the ferroptosis is one of the critical factors in MASH development. So we thought VOLs can be used as non-invasive MASH biomarkers. As an animal model, we used diet-induced MASH model mice. In this model, the pathological features of MASH were observed from the 3 weeks after starting this diet. So in this experiment, we fed mice with this diet for 3 weeks to create the MASH model mice. And we also examined the group fed with CDHFD supplemented with vitamin E. This is a lipophilic antioxidant as the ferroptosis inhibitor. The ferroptosis signature increased in the MASH model mice, and this is suppressed by the vitamin E adding diet. And we also found that the VOLs in biogas and liver tissue of MASH model mice increased as similar. Surprisingly, these VOLs could not be detected in the other tissues. So these results suggest that this technology can also be applied to the non-invasive detection of the chronic liver diseases. Then we have conducted clinical trials. So in this study, we aimed to determine the ferroptosis biomarkers elevated in the exhaled breath of patients with MASH. The breast biopsy analysis was performed on the healthy individual and the MASH and the liver cirrhosis patient. The VOLs in the breast of patients were collected in the thermal desorption tube using the breast biopsy collection station. They're located at the 2 medical centers. Then these tubes were transported to the Kyoto University for the analysis using the TDGCMS. Then, as a result, as expected, the level of the 2 ferroptosis marker was significantly elevated in the exhaled breath of patients with MASH. The level of the 1-10-3 was significantly higher in the patient with MASH than in healthy individuals. However, this increase was suppressed in the liver cirrhosis group. In contrast, the level of the 2-pensulfan elevated in both the MASH and the liver cirrhosis group. Then we also analyzed the correlation between the biomarkers and the clinical markers obtained from the blood biochemical data. Importantly, the 1-10-3 was significantly correlated with the level of the ALT. On the other hand, interestingly, in the MASH and the liver cirrhosis group, the 2-pensulfan showed the moderate correlation with the fibrosis, the marker, the FIFO index, and the M2-BPGI and the liver function marker RB score. So these differences might be due to the chemical properties of these markers. The 2-pensulfan have higher lipophility than the 1-10-3, and so it could be accumulated in the liver tissue for a long time. Also, 2-pensulfan is degraded by the P450, and as P450 activity is decreased in the liver cirrhosis patient, the accumulation of the 2-pensulfan may be more pronounced in these patients. Also, the further detailed investigation is necessary, but these results suggested that combining these two biomarkers could be useful for the diagnosis, the MASH and the liver cirrhosis stages, and identify the patients. And finally, when we combine the ferroptosis marker with age, and we are able to discriminate between the healthy individual and the patient with MASH and the liver cirrhosis with high confidence. I'd like to summarize the main findings of this research. In this study, we employed a bottom-up approach based on the in vitro experiment and identified VOLs, especially released from the ferroptotic cells. Furthermore, using these markers, we developed a technology for non-invasively detecting the ferroptosis in vivo. So our developed markers and the detection technology will be utilized to the non-invasive and early detection of the ferroptosis-related diseases. This is the last slide. I'd like to acknowledge the people who worked with me on this project, and thank you for your kind attention.
Thank you, Yuta. That was a really interesting talk, actually, a good demonstration of translational work going from cell culture to animal models and then into human subjects, and interesting to see that activity related to ferroptosis and also the activity related to CYP enzyme activity in the liver, which mirrors a lot of our own work here at Elstone in terms of looking at liver disease and liver cirrhosis. So fascinating talk. Unfortunately, Yuta won't be able to join us for the Q&A, but any questions that have been put in the Q&A box, we'll make sure that we liaise with Yuta and have those answers available in the next few days via the Breath Biopsy conference portal, where you can also catch up on the talks that you're unable to attend live.